today I can make it with all of you here. And for today's topics is not to say new, but is an important topic, quite was active or very well been debate in 1990s. But for currently with this new kind of industry, herbal pharmaceutical industry, so we have to, I think, relook again on this non-genotoxic carcinogen. So my aim is that just to bring everyone to think together regarding the safety issue on our herbal pharmaceutical product. And I try to bring out a few issue, a few problem statements, and then try to highlight what is genotoxic carcinogen all about, and also the model of assessment to determine the carcinogenicity, especially in vivo model. So I try to be about 40 minutes in, okay. So number one, this I think everyone who are working in biohealth product know the step clearly here. We start with in vitro and then in vivo, and then if we are success, we can go into clinical study, clinical trial, and to commercialize the product. But however, I think we have, as a scientist, my advice, or advice to myself, okay, I think already served university for about 12 years. So as a scientist, as I put in the notes on the left side, what are we looking for actually? Is it for 20 years we're going to work on in vitro to see that mechanism, this mechanism, until when? And then we also, if we success, we can go to animal study, and whether we can commercialize it, or do we just focus on fundamental and we do not want to make use of it. So this is another dilemma of our young scientists, where we are and where we should go. And when you talk about the herbs product, how are we going to determine the safety and also effectiveness? And we are working on it, but until when? This is the thing that we should think of. And at the end, people are going to use it, and especially our medical doctors, okay, are they confidently they can use it. So these are, I think, for me, the number one problems, and especially for herbs research. I think through my sort, we already organized few conference, and I believe everyone agree with me. Majority, I can say about 90% of students or researchers presented their work mostly on herb study, natural product. So, but when I attach with this ministry mostly, so we are looking forward at which stage, or what are the product that we can bring out there. I had few debates with KKM, with IMR group. They said there's a few product, but I'm not really satisfied, or we want to see the evidence base. So that's why that I highlighted a little bit here, especially the non-genotoxic carcinogen with regard in our natural product research. Second thing is about, we have to understand as well, because many people study in vitro and then in vivo, and to commercialize it. But in terms of data that we gain, I just highlight that this in vitro, you cannot simply make a formula if you find in the from our cell line work, and then we can convert directly to in vivo, and then convert directly into human or in clinical trial. Okay, then they're not such thing that straightforward. But we have to do it, we have to perform it, and to gain some data, a brief idea. Is it safe enough with this kind of dose? Okay, but in clinical trial, there's a different thing. So all this data we need, but it's not that put in our mindset that the dose that we should work is according to what we gain from in vitro, and then we convert to in vivo. So this is an important thing. Um, with regard of this cell line work, I think I just highlight little bit more detail of this misconception, because sometimes we use different science, the different, and also the target organ is actually in different condition. Okay, and then the understanding of cell type also, the students nowadays I think should really clear what is fibroblast that different from epithelium, and also the susceptibility of the cells and compare to the target organ. Okay, so the systemic model in animal study is important. As I shown here, another example, if you talk about you doing work on cell line, on prostate cell line, in human prostate cell line, and if you, and then you run in preclinical study in vivo, so the tissue, the anatomy of the tissue or the organs totally different from human being. So that's why I just highlighted here that you should understand with what is going on. Now, the another problem statement is that within 5 years ago, or maybe 3 years ago, there's a big debate about aloe vera, okay, in FDA. What they found is that by using 2 years of rodent model, okay, 2 years assay, we call this 2 years assay model. So they give the aloe vera product, and what they found is that this aloe vera is carcinogen to create intestine cancer, okay, intestine carcinoma. But somehow there's a big debates. One group say in this way, another one try to defend with whatever. So you can go along, have a look on it. But what I'm trying to say is that in our Malaysian product, okay, how are we going to know, how are we going to determine, okay, who going to work on it? So for your information, these 2 years essay, it will use about 500 to 800 rats, okay, and then only then you can confirm whether the product is a carcinogen or non-carcinogen. So you can see, even at beginning, one of the one, not one, actually 3 or 4 compound of aloe vera being found as a mutagenicity in a cell in an in vitro study. But then people still waiting for the animal study. But after 2 years, so it's conclusive to show that there is an carcinogen activity. But there is some big debate on it, because they are pointing out from where, what type is it, the latex, or they blind to, they, put blend together. So all this was bring up. So but anyhow, as Malaysian scientists here, and we are working in toxicology, I think we should alert and know what is going on, and how about in our local research, products going on. So this is the thing. And with current condition, now people talk about black ginger and also cannabis plant. So everything we have to think two things. One is about effectiveness, and most important is about the safety, and also for the long-term use, whether it actually work as for the treatment, but or maybe the side effect, or maybe worse, it become as a carcinogen for us. So this is 3 kind of problem statement that I bring for you to think of. Now, regarding non-genotoxic carcinogen, before we talk about non-genotoxic carcinogen, we should understand the carcinogenesis process. Okay, normally if we learn pathology, so if we know this formula very well. IPP, okay, initiation, promotion, and also progression. And this is related with an activation of a tumor suppressor gene. All this because of the modification at the gene level, okay, at the all the genes will be modified. But however, with this non-genotox, that it might also cause a DNA adapt. So it bind to the DNA where some assessment cannot find the defect or the modification at this kind of DNA. So DNA adapt is one type, okay, one of the mechanism that involved in the non-genotoxic carcinogen. But there's few others more where I cannot. Maybe in next time, if we have time, we can go on in detail. on this genotoxic carcinogen. Here I try to give little bit brief on carcinogenicity. Okay, about this non-genotoxic carcinogen. Normally, carcinogen test is to determine the tumorigenetic potential, okay, in whatever product, and also during over the period of major portion of the lifespan of the test animal. And normally, carcinogenicity tests be conducted if the data from other source suggests a tendency for tumor induction. So we have to clear what kind of data from other source. What kind of other source? Is that the IC50 from the normal cell line? Is it the clonogenicity that we done, or cell proliferation assay? So which data that showing a tendency, or is it the Ames test, or whatever the thing? So this is another issue. Okay, in addition, both carcinogenicity and also chronic toxicity we can do in one single experiment. And the mode of action of carcinogen in two ways. Okay, genotox and also non-genotox. This is notes from the IARC in France. The one popular in the world nowadays, not nowadays, actually from old time, is in France, in Japan, in Korea, of course, United States. So these are the countries who are very focusing in terms of the mechanism of the non-genotoxic carcinogen. Not many countries study these non-genotoxic. Many people are focused on genotoxic, but more, I think, more harmful and more dangerous is that is non-genotoxic, because we cannot determine in normal assay. So genotoxic carcinogen normally increase the tumor frequency in animal cancer bioassay, and also positive in vitro and also in vivo. So this is clearly, we know this is a genotoxic. Okay, or from other kind of analysis, we also can do, we can determine is indirectly acting as a genotox. However, for non-genotox, it usually acts a tumor promoter. So if you know the basic of the carcinogenesis, so the promoter play a critical role. So somehow these chemicals or product that react as a tumor promoter directly, okay, and normally positive in animal study, but negative in genotox tests, okay. And the mechanism, there's must a few mechanism pathway through chronic injury or hormonal mechanism. Hormone mechanism may be the majority of it. Some of these non-genotoxic carcinogen play around, and have a kind of DNA adapt at this hormonal particle. And also, this non-genotoxic carcinogen will increase the cell proliferation or decrease in the cell death target organ. But we also have to put in our minds that not 100% whatever that we study, as I mentioned earlier, what we find in animal that will relevant to human. And there is a few example, okay, especially in the liver cancer in rats, and also kidney tumor in the rats, and also thyroid tumor in the rats. It's not exactly mimic in human condition. In our last conference in MyCourt, our keynote speaker have already talked details on this particular issue. Normally, tumor are seen in animal tests only at high dose level, okay, and also severe toxicity. Whereas in human condition is a little bit different. And in animal strain that we use is known to be especially susceptible to the tumors. So to say that this animal model is mimic to human is another issue. So that's why we have to really understand with what we are handle, okay. So this is a common kind of figure where in Japan, in toxicology lab, they are using this particular kind of figure. If the genotoxic study that you done either is yes or no from the in vitro, you should go, Okay, I'm using a kind of Japanese style. You shoot all the time. They use shoot. So you should use this particular model. Either your genotox positive or negative. You have to confirm, double confirm in animal model. That is what I was learned and been trained. So we have two kind of model. Normally, we start with the liver organ first. We call medium term carcinogenicity. Only target on the liver. Okay, we do some special dissection at the liver, and then we cut off the liver, two-third, okay, hepatectomy. We remove the liver, and then we give the rats with our substance plus with the promoter, and see whether whether the liver becomes CA or not. For your information, liver is the most proliferated tissue. Very easily it can be proliferate. So once if you put the promoter, and if your substance is a causing cancer as a carcinogen, so if we will develop the liver very fast. Okay. So within 2 months with 120 rats, we can confirm whether the product is carcinogen or not. And then we can go for the next model we call long-term carcinogenicity. It take about maybe 5 to 6 months for 5 more organs. So this organ is alternative for what FDA have their model for the 2 years model. So FDA know very well about this. We call this Ito model. I show you after this who is Professor Ito. So he the one who create this model. Okay, and being recognized by FDA, and a few groups in Brazil, in Japan. Of course, Japan the one who start this model, and also Korean, and also Thailand, France. Okay, so a few countries they start to use this liver carcinogenicity study. No, sorry, and also with the long-term carcinogenicity for 5 organ, and then we can make a good conclusion. So this is Professor Nobuyuki Ito. He already passed away a few years ago. I have chance to meet him. I was trained under his group. Okay, he the one who create this particular model. Okay, it's very well known in the world regarding in this toxicology society. So this is example of the first the early papers published by Professor Shirai, and also Professor Nobuyuki Ito, and also Fukushima Sensei. Fukushima Sensei came to our MyCOT conference before, and also present regarding the threshold of the non-genotoxic carcinogen. So Professor Fukushima is already they are more expert, and they already more go more forward until talking about the threshold, about the threshold dose, about the non-genotoxic carcinogen. So these are paper where they already test about 291 chemicals. Okay, and the left side there you can see Professor Tomoyuki Shirai is my professor, where I was trained under him. Somehow, I feel sad. He also already passed away about maybe 2 years ago. So I feel a little bit sad with my professor there. So anyhow, they found many chemicals. So these are the model that been used for the in vivo carcinogenicity. Forgot to mention that we also have in vitro carcinogenicity according to GLP method, that you can also start off before you do the animal study. So in this model, you give a DEN as a promoter for 2 weeks, and then you give your substance, and then actually you have to do at that before that you should do the two-third partial hepatectomy. Okay, that is very quite complicated, should be trained well. There's a tricky thing how to do that two-third partial hepatectomy, and after that the rat still alive, for about another 6 weeks. We give our test chemicals, and then we do, and after we sacrifice the rats, we take off the liver, and we run the GSTP immunochemistry, and we can determine whether it's a carcinogen or not. As you can see here, on the A is a normal, okay, B also the one with a tested chemical but very low GSTP. GSTP is one of the marker for liver CA, and you can see at the C slide, the liver is more enlarged, and you can see highly expressed of GSTP marker. Okay, so that is confirmed there is a carcinogen that been tested. This is another model, multi-organ carcinogenesis. We use five of promoter and then carcinogen. So to target five more tissue, and then within 6 months or 5 months, we can determine whether our substance is a carcinogen for that particular tissue or not. Okay, this is a quite old model, but until now been used, okay, as an important model in carcinogenicity. So these are example, okay, of new non-genotoxic carcinogen. You can see the M is the mutagenicity assay AIMS test. If you understand, I think maybe everyone maybe know about AIMS test, where we use these microbes, bacteria, and as an assay to see the point mutation. Okay, but then when we compare this AIMS test with the carcinogenicity, you can see how about for these chemicals 200 over or 3, now I think maybe only 400 or 700, that showing AIMS negative. Somehow when you give to this animal model, so it become positive, it become carcinogen, as you can see here. So this is what we call the non-genotoxic carcinogen. Okay, and here also there's more others. There's many kind of chemicals out there. So here again, so but in our pharma product, okay, in our herbal pharmaceutical product, I'm not sure. I tried to get the data, but I couldn't find much. When we talk about aloe vera, actually aloe vera is genotox. It's already showing mutagenesis. So, and many groups, they are focused on chemicals, okay, not the herbal product or whatever. So that's why I put this particular topic to our society, have to think of, and how whether we can start to look on this or not. And although traditionally the herbs being used, but be afraid also, if we don't do a proper evidence-based study, we might also get some negative results. So I think that's all regarding my talk. Sorry, maybe I go too fast, but maybe we have time to discuss. And I also open to collaborate with anyone. If you are interested to do in this preclinical tox study, you're welcome. And also for the in vivo, if you feel to be trained or you want to do this kind of study, you are welcome. And also to do the effectiveness study, especially on the cancer model, you can come over here, or I'll come down there also. It's also possible. And with that, I think thank you, and I'm free to get any question. Try my best from my understanding. I try to help you. Okay, is it okay? Am I too far? Is it?
Thank you very much, Dr. Azman, for your sharing. Now is Q&A session. So please post your question if you have in the chat box, or you can unmute yourself and introduce yourself briefly, and then throw out your question or any comments or specific project, or anything that you want to say to Dr. Azman. Any questions? Dr. Azman, if they don't really have any question, I would like to ask first though.
Yeah.
Okay, when you're saying about the model, right, the in vitro and the in vivo, people always go for in vitro in terms of to do the fundamental part. And I think a lot of grant application now, I think they are lacking in terms of commercialization as well, right?
Mm.
Especially related to natural products. So, do you think that like focusing on one natural product and then do a complete study, like do fundamental, do the preclinical study and do commercialization is the best strategy for the research to go, or just do like screening for multiple natural products to know like the effectiveness of each natural product?
Yeah.
Maybe you can share your opinion.
Yeah, yeah. There's a few things. Number one, we have to ask our own self what we are looking for. Although there's some limitation on the grants part, but as a scientist, as a lecturer, as academia, we have to ask ourselves what we are looking for actually. Do we looking for certain proteins, genes through our study targeted, we want to show off this particular genes or proteins, or we want to show off the plant itself, or we want to study to show that we have create a model. We are good in modeling to give a model in terms of in vitro or in vivo. So that one, we have to ask ourselves what we want to profess in, what we want to be expert in, or what we are looking for actually. So that is number one. Number two, if we say that, okay, I want to get this plant, to this plant showing very good kind of treatment from my whole, my study in vitro is showing a promising evidence is there. So if you, then you should go move forward, even though it's little bit difficult, but there is. Regarding the grants, I don't think much that is lack. There is a PRGS grant, even at the ministry level. There's cradle to commercialize it. There's a few actually. For me, from my understanding, actually more, but we are not being being info, or we do not get the good information regarding this. That is one part. Another thing is that with the industry, if we have certain plant product or herbs product that we think can commercialize, then we have to go out with the industry, make partner with them and make friend with them. So how this product can be, get along with them, they can they can funding it, they can give the money. So that is the thing that we should go move forward. Or as I said, that we want to create a model. We are good in this, in certain assay, enzyme assay, to confirm whether this is, you can determine the carcinogenicity or genotox. So we are go deep in that particular thing. So depend. And for me, it's not a proper if we just get along with what government provide the money. You need the money, but at the end you have to ask yourself, of course. And sometimes I think in some condition, better we balance in fundamental and also application. Some people out there, some professor, they think we are should be very good in fundamental. Is correct, but until when? And what is the end point, or what is our target with our fundamental? Who going to make use of it? If we the one who find we do the fundamental work, at the end we also cannot use it. So how? So we, the money that being given to us. So why not? Not only why not, but we somehow we should also, go on with application. How to make use of it in term of simple way, in term of a more advanced way. So it depend on us actually. Everything depend on us. What we are really target. So that is my personal opinion through what I've already gone through. So regarding whatever plan you want to screen, I think many institution, we already more than 20 years, I think more than 10 years, many institution already screen many kind of plan. They've done a lot of in vitro study and go so on with it. But when we go to another step, preclinical study or clinical trial, then we are lacking of data. If I'm not mistaken, I just today's I have some literature if someone interested. I found that even from our Chinese traditional medicine, uh, people have done a survey. Still, the lacking data about 90%, 80 to 90% lacking data, a good data for clinical study. So that's how that I think there's many gaps in this kind of research that we have to strategize and to move forward. At the end, we have to ask ourselves in which area, in what condition that we want to be professed in. Today we take one plan, tomorrow we take another plan. Okay, so at the end, I'm not sure what we are looking for.
All right, thank you, Dr. Azman.
Yeah.
There's another question from the audience. Prof, is there any limitation of doing toxicity by using zebrafish, since the organs of the fish are not as big and not complete as rodents?
Yeah, of course there is some limitation, but what we are looking for, okay, because there is advantage, pro and cons with these fish, okay, zebra fish, because it's small, you can do a lot, and also some of the target tissue is more mimic to certain condition of human, so then you can make use of it. But when you talk about clinical trials, so you have to know what are all the other data that also be needed. But if you want to put as a screening material, or do you going to talk about the embryo or whatever?
Eh, seems like Dr. Azman left the meeting. Dr. Chan, maybe you want to answer that question. I think it's a question um, by one of the participants. Maybe you can share your thoughts in using zebrafish, Chan, while we were waiting for Dr. Azman to come back.
Yeah, I don't have any experience using zebrafish, but however, there are actually a lot of these researchers out there right now using zebrafish
Mm.
as a model. And what is right is actually about what Prof. Azman has stated just a while ago was that you have to look at the objective of your study, all right? Because definitely zebrafish has been shown to be around 70% of the genetic composition is actually similar to humans, homolog, all right? And has been shown in embryo toxicity as well using zebrafish, yeah. And however, this has to be confirmed again. I mean, we confirm for several tests because one is just for to demonstrate. The other one is for confirmation, right? I think it's still doable for screening purpose.
Okay. Chan, unfortunately, we are still waiting for Dr. Azman. Can you help to answer questions coming from the audience?
Dr. Razina, can I add in?
Can. Please,
Okay.
please Dr. J.
Okay. Sorry to
So Jangkir,
interrupt.
yes, good.
Okay, my name is Dr. Jahangir. I just want to further reaffirm Prof. Mahathir, Dr. Azman, and also Prof. Mahathir, Dr. Chan information regarding zebrafish. Just to share to everyone, the OECD itself, for the regulatory purpose, have recognized certain parameters for the purpose of to reflect the mammalian toxicity. So yes, it is not the whole human system to reflect to predict on the toxicity, but actually OECD themselves have already identified. So therefore, it's not only at the level for some parameters, not only at the level of screening, can also be used for regulatory purpose. So therefore, please refer to the OECD, relevant OECD test guideline that use zebrafish. Thank you very much.
Okay. Maybe Chan or Dr. J can help to answer. There's another question
Hello,
from Dr. Asma. Ah,
hello.
Dr. Azman, you're back.
Yeah. Sorry. I'm
Ah,
not sure what happened.
okay.
Okay.
Dr. Azman, are you okay
Yeah.
to answer another question?
Yeah.
Okay. There's another question on what is your opinion on some tablets capsule that has been developed from natural products and given to subjects just based on the traditional usage and preclinical data. So it's based on in vitro and in vivo study only.
I think whatever the thing, they should go an ethical approval. If you use a clinical model, a human trial, you should gone through the ethical committee. So the ethical committee and also the research committee of the institute already gone through, and they should pass over all the important data through the in vitro and also in vivo. Of course, the data, as I mentioned just now, there is a basic information from there. But we still need the data. But to translate direct from that data to human clinical is impossible. So whatever is it, there is a body authority from the the human ethics. They are the one who will go through the data and to approve it whether it can be can be conducted or not.
All right, Dr. Azman. Thank you. So there's another question here. What is the suggestion for toxicity tests of herbs with the limitation of budget? So meaning that we want to test the toxicity, tapi we have limited budget. So what kind of testing would you suggest?
Budget, number one, somehow you should find the budget, okay? And we cannot use the excuse to, okay, because we only have this small budget, so we have to do this particular kind of model. So that one, definitely you have to find, either from the government or from the industry part. But to say, normally we go for screening. I think everyone know about L929, NTTSA or whatever. But for me, that is not enough. And normally, if you have small budget, you can use multiple of cell. So try to work with a few cell and the one who are close to the target usage. Let's say you're talking about liver, you're going to work on liver CA. So then you find a normal liver as a comparison with it. Don't use mouse fibroblasts and then you're talking about liver CA. So both mouse fibroblast is a one step, okay, it's number one, but you need others, and also try to go more deep. If you not have so much budget, try to go more deep and see what actually this plant target on which particular genes or proteins. At least you have some data how the mechanism, the working, the act of this particular agent.
Okay. All right. There's another question here. Is there any herbal pharmaceutical product in Malaysia that already passed clinical study?
From what I got information from IMR, the one who are responsible for this particular exercise. They say yes. But for me, I'm not sure what is it. Because I don't get the actual, but I believe maybe there is yes, as what IMR claim, especially the industry. In my side, I'm working now with Anta Pharmaceutical. So we are working at preclinical, and if good, then we will go for clinical trial. But IMR, the MPRA already set up a new SOP for the herbal usage in for the treatment. They just released the protocol.
Dr. Azman, in relation to that question, right? Do we need like all these pharmaceutical product, all these herbal product, do they need to submit toxicity tests? I mean, before commercialization or before?
Yes. Whatever, yeah. You should go to get, even if you're going to register, even simple NOT, you should submit this toxicity study. I have, just to share some experience, I met Prof. Bay in Duke in US, Singapore. He has 3 papers on Nature, where to show the sleep from there's a chemical from slimming product, herbal from China, and this cause toxicity at the kidney. And with that paper, 5% of the pharma Chinese product already gone down. So so it's very important, to understand with what we are working.
Okay. There's another question. We have a lot of questions today. Good. So some say it is safer to drink herbal tea from herbal powder rather than eat as ulam. What is your opinion on it?
My opinion, we follow if a traditional way, and normally we take ulam lah, we take. That is been practiced, but the most important is the evidence base to see the safety or not. Like recently I've been told that red spinach, bayam merah, is very toxic to cause batu karang. I try to look over the data whether it's true or not. So somehow, it brings that for us to do a study on it. So whether powder or the water, it depends on the solubility and also the bio ability of this particular product to absorb in our body. That one you should do a study. But if you ask traditionally, we have to ask the traditional user how they make use of it. And it's good also when you do study, you try to see almost similar with what the people out there use. If they boil and they get an extract through a boiling system, but you do petroleum ether extraction, blah 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 extraction, and at the end when you're going to commercialize it, you have big trouble because to set up that particular thing is another issue. It's very expensive. But if you're looking for active compound, it's different. So you can do whatever extraction because you're going to study the active compound and you're going to isolate it. So that is a different issue. But if you're going to mimic whatever the traditional way, so we have to do the same thing in our experiment.
Okay. All right. Any other question?
Hi, Dr. Azman. Hi, everyone. I'm Rosaini from UTM. So I'm actually working with genotoxic carcinogen in herbal products. It's quite tricky when we deal with natural product or herbal medicines. And one of the big issue I think is about the mixture effects of the product itself. And usually, Dr. Azman, how do you start when start a research about the traditional medicines or any supplementation? Okay, on one product, on one basic, one main ingredients, or you do the mixture effects?
No, for me, I go more mimic to what the traditional usage. More simple way, just by boiling, I start with that one. So I more love in using crude extract rather than the active compound. And because what I believe is that all these should work together. They have a synergetic activity among them. All these compounds actually mostly available in all kind of plant, but then they have certain group where other plant don't have. So this is what my own opinion. So therefore, it's good for me to use a crude more in simple way rather than going for the active compound. But you should know what are the compound. That is should be because you want to know with what you are dealing, but not to isolate it. Isolate is a different. So like our vitamin E, tocopherol, tocopherol, also there's also issue on it. We need the natural one rather than synthetic. It's very difficult to synthesize all these compound. It's not everything can be synthesized.
True, Dr. Azman. Thank you so much.
Yeah.
You start with the crude and then you focus. Once you know the effect, then you focus.
Yes.
Know what's actually inside the product.
Yes.
Thank you, Doctor.
Yeah.
Any other question?
Chan here.
Okay.
Yeah.
Okay, all right. Dr. Azman, just need to ask you, based on your experience, how many the rodent carcinogenicity assay has been conducted in Malaysia, and what is actually being tested on? Do you know?
No, in Malaysia, I think the carcinogenicity in vivo, I think no one have done it.
No one has done it.
No. In Malaysia, maybe none. In vitro carcinogenicity might be, might
Yeah,
be.
in vitro, yes. There is.
But not the genotox a lot. Genotox in vitro AIMS test, I think a lot.
Yeah.
But cytotoxicity as well, everyone doing it. But when you come to carcinogenicity,
No
I
one.
don't think so. Yeah.
All right.
Toxicity study, yes. Toxicity, acute tox, that one a lot people doing it.
Okay. Dr. Azman, we have two more questions to answer before our AGM.
Yeah.
So nowadays, it is a lot of product that claim that they are good enough to treat certain diseases
Mm-hmm.
from A to Z, especially a product which can be used in reducing fats. What is your
Yeah.
opinion? I think like a poly pill or a poly capsule which can be used to treat everything. So
Mm.
what is your opinion on it?
Yeah, as a scientist, we need evidence. So that is
Yep.
number one. But
Mm.
when you come to this group, to this industry, to this, these companies, they like to put everything, such as that particular product as a god, to treat every kind of disease. So without just by using the testimony, and this is our role, I think, as a scientist. We should make the awareness about the different of testimony and also research evidence. So if you see clearly out there, many products they are using testimony, patients
Mm.
testimony, which is totally wrong. 1,000 testimony is not equal to whatever one paper, our finding, you see. So this is the dilemma, or the in any part of the world, people who are having money, they start to do business and while claiming, or so it make worse to user and also the community to believe. So that is our role. I think we have to sort out and to show. That's why we have to take all this product, do research in proper way, and help join with the particular industry to bring out our national product in a good way.
Okay, Dr. Azman. I think last question, both are actually the same. So, Prof. Azman, what is your personal opinion about venturing into cannabis or ketum research as therapeutic agents in Malaysia? Any current legal challenges, and whether do you think it is feasible to do about ketum?
It's really great question, and also target to my head. I also joined. We try to make a society, my HAM, Malaysian Medical Ham Society. I was in the committee as well. So I talk about cannabis first. I think ketum everyone know, and actually been practiced by our elders. But now the youngest make make use of the ketum in different way. So that is the worry about the ketum. Maybe there is a positive, but as a scientist, I think we should work out with a good formulation, with a good dose, how this ketum can work out. I believe on it. I think many people already done study a lot, foreign and also in our country also. We have friends a lot, people already study on ketum. But how to formulate it to bring in a good way, okay? But we have to control the growth of the plant, because people misuse it. About cannabis, it might more interesting, because the use of it is a very, if you make use in a good way, is definitely very good. But government should control it. And until now, although we already changed the government quite a few times, it's still not move on. There's a group already work on it. They already have a plant area where to move, to plant the cannabis, but still at very beginning stage. But in the worldwide, even Pfizer company already invest in this cannabis. We have to know about cannabis, the THC and also CBD. CBD is much more, the one is good compared to the THC, but THC also at very low level also can work out. Um, I'm not sure how many of us that understand that we are using morphine, we are using uh, all these other drug for disease. Okay, for there is a patient using morphine or whatever the things. So if you compare this one with what THC and CBD have, is much more better. But again, we should clear up all the thing that need for the safety and also effectiveness. From my understanding, United States, whatever they use the CBD and THC, none of them died because of the high dose or whatever. So that is the one part of advantage. I think I cannot explain much, because there is, as what Chan mentioned, like COVID, many people out there now following the Google's professor. So Google professor give many more ideas, and we also maybe believe on the Google professor. And if someone behind the Google professor, so all this propaganda will come out, and many things. But I think you should go by yourself, study, and you are a scholar, so you open your mind, and there's many information in the Google, in the YouTube, in the papers, scientific papers, from the FDA also. They already have come out with all this, whatever the study been done in the whole world about cannabis there is, and you can see how much good or positive or negative about it. But in Malaysia, we cannot touch it yet, although the issue is that they're going to change in the undang-undang akta dadah tu. Dia kata jawatan penjawat awam. We are not penjawat awam because we are in the university, we are badan berkanun. So penjawat awam ni siapa? Polis, tentera, jabatan hutan. So they are not keen to do research. So these are the conflict. One of the conflict why we still cannot do. But I think maybe FRIM and under the CAS Kementerian, whatever the CAS Kementerian and the FRIM, they are doing something. Yeah.
All right, Dr. Azman. So thank you very much. I believe that people who want to work with Ketum or cannabis, like they can, of course, they can email you or they can collaborate with you. Do you think?
Yeah, sure. Let's go. We have to go. But never mind. You can PM me. We can chat. We can chat in a long way. Yeah.
Yes, of course. So thank you very much, everyone. We are actually right on time because 4 minutes to 10 o'clock. So thank you very much again, everyone, for participating actively in this special toxicology webinar series organized by Malaysian Society of Toxicology. So on behalf of Malaysian Society of Toxicology, we thank you guys for your participation. And I have posted link for our AGM only for invited members, for active members. So if you guys would like to join as members, you can do so and visit our website. But I would actually encourage everyone to join our society to get more information and to join our activities related to toxicology in Malaysia. So with that, we end the session and we go to the next session for the AGM. Annie, do you want to say anything?
No, thank you, Dr. Razina. Thank you, everyone. Thank you, Dr. Azman, for the talk.
Yeah,
and
thank you.
Okay, thank you, everyone.
Thank you.